Hi, everybody. I'm Sue Tappan, a member of the SPARC Data and Resource Center. On behalf of SPARC and the Codeathon mentors and judges, I welcome you to the 2023 SPARC FAIR Codeathon winners announcement. This 3-day virtual Codeathon ran August 5th through 7th and was made possible with funding from the National Institutes of Health. In its third consecutive year, the Codeathon invites teams from around the world to come together to explore and build something new. SPARC has a lot of unique data, extensive access APIs, and powerful processing tools. But first, a brief introduction about what SPARC is. So SPARC is a common fund program funded by the National Institutes of Health that is focused on bioelectronic medicine and neuromodulation therapy. SPARC stands for Stimulating Peripheral Activity to Relieve Conditions, and it emerged as a research focus because neuromodulation is an emerging therapeutic approach to treat diseases of the autonomic nervous system. There is limited understanding, however, of which neural targets to influence and how to precisely control organ function. Therefore, SPARC is charged with the efforts to deliver detailed, integrated, functional, and anatomical maps and models of the peripheral nervous system and its innervation of target organs to bring the power of convergence across molecular medicine, neuroscience, engineering, and computational science to develop devices to diagnose and treat disorders. The SPARC community is created of researchers working across the peripheral and autonomic nervous system. There are over 250 curated data sets and computational studies containing multimodal data, physiology, morphology, anatomy, transcriptomics. These have been contributed by over 60 research groups, at least, spanning 90 institutions and companies and working across 15 organ systems and 8 species. As I said, the Data and Resource Center from SPARC creates the infrastructure for sharing and using this data and provides other publicly available resources. We provide data management and publishing, computation and simulation, knowledge engineering, multi-species maps and frameworks, and manuscript and protocol links to ensure that people can understand and explore the data that is present. So the SPARC portal is an integrative portal that features high-quality data sets that span species, formats, methods, and ages. They go across all types of modalities, across numerous organs, and a diverse distance as well. Regardless of the modality, species, or format these data sets are created from, on the portal, they have a consistent layout, explanatory text to help you understand the information and the modus operandi for why this research was undertaken, and the data is presented in an accessible way so that you can use it, reuse it, learn from it, and establish collaborations. For longtime members of the SPARC team, it is exciting to see how people with fresh perspectives on the project and platform want to use the platform to do real science. While our goal is to make that process ever more convenient, we require assistance to understand where the gaps and challenges exist. Identifying these gaps represents opportunities and sometimes some frustration. We appreciate everybody who was willing to give up their weekend to live that in real time. Each Codeathon project must use SPARC data or SPARC tools and resources in novel ways, particularly in enhancing, demonstrating, or measuring the fairness of the data or tools and resources. Each project is judged based on the creativity of team solution, impact on the SPARC research community, feasibility on the prototype implementation, as well as completeness of the documentation. Let's celebrate each team to see their project and solution before we reveal the outcomes of the judges' deliberations. First up, team 1.
Thanks for joining me. My name is Jerry Skeffis, and I'm with the MetaCell team. During the Codeathon, to develop a tool to integrate two different pieces of software so that you can visualize from SCAN in the Open Physiology viewer. And so what does that mean? SCAN is kind of the pathway at the bottom here where you're taking models that have started as NLP models, and then you've got expert curators that are working to refine those neural network models and put them into a database. But then app anatomy is a methodology for expert curators to create a circuit diagram showing all of the different connections throughout anatomy so they can represent physiology in an anatomical context. And we thought it would be really cool if you could take these models, all of the models coming from this SPARC connectivity knowledge base, and represent them in app anatomy so you could learn a lot more and explore how they work. So this shows the tool Scan Composer that we've previously been working on, and we have ongoing projects to develop. And then this makes very simple point A to point B network diagrams, and you can actually expand those over time as we continue to develop the tool. App anatomy, on the other hand, has been going on for quite a while and has a lot of different complexity that you can add in there. So you can add multiple scales of anatomy coming from organs down to tissues and cells and subcellular locations, and the same schematic can be used across all sorts of different species and across all sorts of different physiological scenarios. So the approach for this project was to take the latest release of the database coming from SCAN, and that comes out as a turtle file that then needed to be converted to a JSON, and in turn converted to a spreadsheet, which is what the Open Physiology viewer can use. So it's a little more friendly for people to create new models. And the asterisks on this diagram show the different components that we worked on for this project. So basically, all of the models that were given to us are able to be imported into the Open Physiology viewer. And I'll show you a quick little video of what that looks like. These are some of the models being brought in, and you can also see that you're able to see it in the context of the whole homunculus of all the different organs. It shows kind of spleen and thymus and things like that. So I'm just going to proceed. This shows also the extension of a model to show a comparison between sexually dimorphic development, because a lot of the models that were given were about the reproductive system. So we showed the female versus the male in pink and blue. And then we provided links to some additional models that were provided. So this is our team. Please reach out with any questions, and we look forward to continue to work on these projects. Have a great day.
Thank you, Team MetaCell. So with that project, it represents one of the first efforts to really delve into our knowledge base affiliated or associated with SCAN. And so we're really excited that it was a feature of and focus of the MetaCell team. The next team also utilized SCAN. So NeuroGroup is team 2. And in the course of the 3 days, they generated some big plans, but were not able to record audio for their presentation. So they did not create a video. These are slides that were created to give you an idea of the application that they were working on, and their code is available on their GitHub repository. As NeuroGroup, Group 2, we really appreciate their perseverance and creativity in their solution. I was quite excited at the complexity and enthusiasm that they brought to the project. They accomplished a lot in a little amount of time, but I think their plans were even bigger than what they were able to produce for us to see. Thank you. Next up is SPARC Flow.
Welcome to our SPARC Flow presentation. The SPARC project provides well-curated datasets for advancing bioelectronic medicine. And what researchers can do is process this data in workflows to generate novel results, insights, and knowledge. However, there's currently no option for users of SPARC data to easily describe their data processing workflows and their tools in a fair manner. It's also difficult for them to run these workflows and reproduce their workflow results. So our solution was to develop SPARC Flow, a Python tool to describe and run tools and workflows for processing SPARC datasets in accordance with FAIR principles. we created a series of tutorials to help highlight the features of SPARC Flow. This includes showing how we can reproduce a data processing workflow from an existing SPARC dataset. And we show how we can describe this workflow using standardized workflow and tool descriptions. And we then show how we can run this workflow both locally and on cloud-based platforms. We also proposed guidelines for enabling FAIR workflows and show how we incorporate these best practices in these tutorials. In terms of impact, SPARC Flow supports the SPARC DSC and community developments. For example, SPARC Flow could be used to describe tools and workflows that can then be contributed to the SPARC portal. SPARC Flow also allows workflows that use SPARC data to run on different platforms. And SPARC Flow also supports our vision for enabling tool and workflow descriptions to be included in knowledge bases such that we can ultimately look towards automatically assembling workflows for different applications. Thank you from the SPARC Flow team.
Days, over 250 commits. Guys, we're really busy. It's very exciting. Congratulations.
Hello everyone. In the SPARC Codeathon, we present SCAN-Compare, a Python package and web app to visualize scan data across different species. Now, what is the project motivation? Before that, let's go to what is SCAN, an established semantic store containing a variety of information. Why SCAN? Because it follows the principles of FAIR. How is SCAN organized? It is basically in the form of connections. Now, what is main motivation? We want to make it easily visualize it so that user can easily visualize everything. So it's always about easy to understand visualization. Now, specific project goals is to retrieve anatomical data, map it to different species, different phenotypes, and then finally visualize it for user to access it. Now, how do we retrieve anatomical data? We set endpoint at place graph, use SPARQL in Python to filter out all the information. Now, to visualize the anatomical data, we manually mapped a basic anatomy of 3 different species, manually assigned different regions to those in the regions in the scan data, and then finally used Cytoscape and Plotly to plot the connectivity data frames. Now, we have 2 different platforms. First one is Compare Python package, and second one is web application. In Python package, it is customizable, portable, and easy integration is there. It is also reusable, whereas web application, it is easy to use, quick visualization, and does not require computational experience. Here is the demonstration of Python package. Here you can see there are different neuronal connectivity for different phenotypes. You can go to any region and it will get you the label of that region. You can go ahead and zoom and you will be able to see that zoomed in area, and you can again go to different areas and have a label of that particular area. We have also a simple representation of selected regions. You can go and select in the Python package the specific region and it will plot the simple representation. We also have projection fibers, which basically says that neuron A project to neuron B. Similarly, on web app, you can choose different species, different regions, and have a simpler representation. You can also have a complex representation, very similar to a Python package. Now, what does this mean for the user? It allows communities who have limited technical abilities and who do not know a lot about anatomy to go and visualize different models. Now, in future, we want to integrate this with SPARC Flat Map. We want to include different species and phenotypes. We also want to include app anatomy, functional connectivity models. Finally, we would like to thank SPARC and SCAN team. Thank you so much for providing us this with opportunity.
Excellent job, Team 4. Moving on to Team 5.
Hello, everyone. So this is our solution for the SPARC Codeathon called SPARC Chat. I'm Ali. My co-workers Anmol Kiran, Jill, and Lee. So just to give you an idea of like what our motivations are for this project, as you know, SPARC portal contains a lot of information across different types, of course. There's datasets, projects, models, studies. So we came up with an alternative user-friendly solution so that the newcomers could get new information more quickly. So this would reduce the amount of time that people spend on the website. So our methods is pretty simple. We used web scraping. Basically, we took some of the datasets descriptions that were available. We put them in the text file format, and we used some large language models for vectorizing the information and also using them for retrieval, and then we managed to find the solution and publish the solution on GitHub. So the requirements of the projects, of course, there's the manual scrapping of SPARC web pages. This could be, of course, automated with APIs, but we had some difficulty with that. We would need to tune the models to get the best performance satisfactory results, and also have users to give inputs through the chatbot interface. This we could use with radio interface. So as a result, we first experimented on Jupyter notebooks, where we had simple queries about the data set. So we had, like here you can see on the example here, where I asked for a study with subjects as pigs, and you can see the information that the model has been able to provide us. And of course, with radio interface, we can make it actually much nicer. You can try it on the browser. Like here, we have like some sample data sets with some like examples of the data set, what kind of limitations that we have. The model provides some answers. And of course, we can ask with follow-up questions like the information about like the links, and also the citations of the data set that is available for rat heart, which is kind of very specific, but we can still ask the model, and it manages to provide us with the answer. So for the future plans, we need to like refine the models. So far, it's only 12 pages of the data sets. We just wanted to have like a proof of concept idea, containerize the installations of some of the packages as they are recent, and the repositories need more stabilizing. So a Docker solution would be much better for this part. And also web chat interface for the SPARC, we could also add it to the dataset, to the website for the interaction with the users. We want to acknowledge the organizers, my members of the team, and also the open source community for all the available code and models. You can try it yourself. It's available in this link for 72 hours. Thank you very much.
Very, very nice. We have one more team. This is Team 6.
Hi, our team represents the project AI SPARC Flows, a tool for fast and small savable workflows for SPARC data analysis. The team includes me, Charles Horn, John Bentley, and Mason Mings. AI SPARC Flows is a dashboard that allows users to input a desired analysis workflow performed on SPARC datasets to do machine learning and descriptive analytics. The requested analysis is used to generate a prompt that is sent to a large language model, in this case, ChatGPT, using the OpenAI API, which in turn generates a Docker file and a Python script file that is returned to the user. The user runs the Docker file to create a Docker image with an appropriate analysis environment and built-in Python script to complete the analysis. So why use this tool? There are two pain points that we deal with for analyzing SPARC data, one of which is fast preliminary analysis, and the second one is to create small footprints of saved workflows, both of which align with our goals and benefits to the SPARC community. Second and thirdly, I want to mention that our approach is lightweight, dashboard implemented in the cloud or locally for the creation of workflow files, including this Python script and the Docker file. And we see this as highly feasible in the sense that we have lightweight files implemented quickly in the cloud, and we also can train the model again, on large language models that might be open source in the future.
The dashboard is implemented with a Flask backend and a React frontend. To install it, simply clone the Git repository, then navigate to the dashboard directory, then run npm install and npm start. For the backend, navigate to the backend directory. Make sure you have the Python library prerequisites installed through pip, including Flask, OpenAI, TikToken, and PyYAML. Then start the server by running python server.py. Make sure you have your Open API key in the directory above the GitHub repository. Set up your local directory structure by having a subdirectory named results and another subdirectory named data. Then go to the sparc.science repository, select your data and download it to the data directory. Make sure the data is in an appropriate format, such as CSV, Excel, or XML. With further development, AI can be used to format the data.
This is an image of our dashboard. We have 4 inputs that we feed to our backend, which then generates a prompt that we feed to our model. Those 4 inputs are the analysis type that we want to run, the file type of the data that we run the analysis on, the included Python packages that will run in the container, and a short data description of the data set that we will be running our analysis on. Once these inputs have been placed, the user will click the generate button, which will then generate the Python script and the Docker file that the user will then download by clicking the 2 download buttons on the bottom of the screen. Once the files are downloaded, the user will place them into a directory similar to the structure that John mentioned previously. And then navigate to that directory, build the Docker image, and then run the image, and then run the container to perform the analysis. The analysis will then, and other generated files will then be placed into that directory, according to similar to the image seen in the bottom right-hand corner. The output and results will be sent to the results.txt file. In the future, we're very excited for the directions that we can take this model. Obviously, we can aggregate multiple data sets into one. We can use open source LLMs instead of the OpenAI, which is not open source, and hopefully train those. Obviously, placing this on the cloud is very exciting to be able to alleviate hardware resources constraints and potentially difficulties downloading and using this tool locally. And hopefully we'll be able to place this in a place that has high availability and will increase the ability for users to access and utilize the amazing data sets that SPARC has provided to the public. Thank you very much.
You all did great work in so very little time. You should all be proud of yourselves and your efforts, and we are too. Given the short time that you had to put together these projects, each team and each member has done exceptionally well. Congratulations. I do not envy the task of our judges. It was a difficult decision to make. These projects were very exciting, demonstrated a wide variety of implementations and efforts, and really demonstrated how groups of people can work across multiple time zones and pull together something pretty exciting and innovative in just a few days. Each team, in addition to competing for cash prizes, are invited to write a manuscript about their implementation for their project. And the SPARC DRC will support their manuscript submission costs. That is available to each team, irrespective of how the judging actually played out. Our judges were truly impressed by the creativity, innovation, and technical expertise demonstrated throughout the event. Following thorough evaluation by our team of judges, listed here on the screen, we're excited to announce the awards. And the winners are: 4th prize goes to MetaCell Does App Anatomy. This repository contains 2 tools hacked during the last 3 days, an updated version of Open Physiology Viewer, and the SPARC Converter. Congratulations, MetaCell! Team MetaCell crafted a visualization tool for 3D app anatomy models integrated into the SPARC MapCore toolset. This project offers a fresh approach to molecular data visualization with well-documented and user-friendly code. A connectivity knowledge base is a novel approach to facilitate the utilization of SPARC resources. This project contributes a systematic tool for visualization with great potential for many different applications. The team's professional coding style and user-friendly step-by-step manual make their tool set simple for other researchers to reproduce, adopt, and revise. Now, third prize goes to SCKAN-Compare. Team SCKAN-Compare designed a tool to compare neuronal pathways across species. This focused on well-documented project holds significant promise as a resource for brain organization research. The team made extra effort to ensure the project is user-friendly. The proposed visualization approach is technically solid, contributing a user-friendly tool for both the scientific community and the general public to build upon. Links to the Jupyter demos and tutorials were very much appreciated, as well as a brief comparison with existing tools. Congratulations, SCKAN-Compare. Second prize goes to AI SPARC Flows. It's a tool for fast and small, savable workflows for SPARC data analysis. Team AI SPARC Flows developed a tool that can generate FAIR compliant workflows for SPARC data. This project can help make it easier for researchers to share and reuse data and workflows. The team's code is very well documented, easy to use, and they've developed a solution that is both practical and scalable. A fast and small savable workflow could potentially benefit many SPARC users on a wide range of applications. The coding style is professional with informative annotations, and the technical document contains a good introduction to proposed approaches, all of which are credited to this great team, with each team member contributing different expertise to this project. The use of a walkthrough with appropriate screenshots to help users use the tool was appreciated. The grand prize goes to SPARC Flow, a Python tool to describe and run tools and workflows for processing SPARC datasets in accordance with FAIR principles. Team SPARC Flow developed a workflow generation engine, simplifying the creation of SPARC data's FAIR compliant workflows. This holds great potential for enhancing data and workflow sharing within the SPARC community. The team's code is both well-documented and user-friendly, coupled with a clear roadmap for future advancements. An end-to-end analytical workflow is critical and novel for bioelectronic medicine. The SPARC Flow project supports SPARC DRC and community development and enables the best use of SPARC resources across different platforms. The work is also well-documented, which has the potential to be published in the near future. The completeness of the project demonstrates good collaboration among team members. This team gave a thorough description of their project for both users and developers. In addition, they gave a clear indication of what problem was being solved, as well as providing tutorials and pointers for the next stage in development. Congratulations. We also extend our recognition to the following exceptional teams: Team NeuroGroup and Team SPARC Chat. Thank you from the judges to all participants in the SPARC Codeathon. Your dedication and hard work is so appreciated. Congratulations, everybody. If you like what you've seen today, before we close, I'd like to alert you to this new funding opportunity to enhance the utility and usage of Common Fund datasets. As a Common Fund program, SPARC has a lot of great data and will continue to grow in years to come. We invite everybody to become a member of the SPARC community and find, use, and share models, data, and resources about the peripheral nervous system on the SPARC portal. Thank you for helping broaden our collective view of SPARC and giving us a glimpse of the different ways that the research community will use our shared platform in the future. The GitHub repositories are freely available and will be unlocked. Manuscripts will be coming out soon. Prize money will be distributed. And we'll all be excited to see what comes next. Take care, everybody.